collapsin response mediator protein 4. Structural features of the C-terminus may explain divergent roles in axon regeneration and degeneration. Presented by Sophia, Bella, Stephanie, and Ava. Presented to Dr. Summer Lee for Biochem 402, Proteins, Structure, and Function. An introduction to the properties, characteristics, and functional role of CRMP4.
CRMP4 is also called DPYSL3, and it's one of the 5 members of the CRMP family. So CRMP4 is a cytoplasmic growth cone-associated protein, and it's involved in things like cytoskeletal remodeling, axon projections, and also cell migration. Although it has dihydropyrimidase-like fold, it doesn't actually have its enzymatic activity, so it lacks the ability to bind things like metal cofactors. But instead, it actually functions mainly through cytoskeletal and protein-protein interactions, especially with actin, tubulin, and other CRMP family partners. But the most important part is that CRMP4 associates with CRMP2 in neuronal growth cones, as mentioned earlier. They co-localize in the transition zone and coordinate actin and microtubule dynamics to drive growth cone development and also axonal elongation. So now we can talk about how CRMP4 actually works in the cells, right? So in neurons, it localizes to neurites and growth cone tips, as mentioned earlier, especially with strong localization in the central and transition zones of the growth zone. So that's where the actin and microtubules interface in both the peripheral and the nervous system. So functionally, it regulates both major cytoskeletal systems, so it promotes microtubule assembly, and it also bundles the F-actin. So crucially, the loss of the CRMP4 protein produces smaller growth cones with reduced lamellipodia and also impair axon extension. So CRMP4 also appears to bind with CRMP2, which may actually potentially form a heterodimer or tetramer, and it bridges the actin and the microtubules in the transition zone of the growth cones. So it basically actively coordinates the cytoskeleton to support growth cone spreading and axonal elongation. Okay, so now that we have a little bit more background on CRMP4, we can talk about one of its main functions, which is cleavage switching. So after an axon is severed, released calcium activates something called calpain, which cleaves CRMP4 and produces a 55 kilodalton trunacid fragment called tCRMP4. So this cleavage site was mapped to T637 within the C-terminal region that is important for F-actin bundling. So therefore, cleavage likely disrupts CRMP4 normal cytoskeletal role. In the proximal PMS axon, CRMP4 stays mostly full length, which preserves the cytoskeletal functions needed for growth cone reformation and also axon regrowth. In the distal axon, though, calcium-dependent calpain cleavage generates tCRMP4, as I just mentioned, and full-length CRMP4 is lost, which is what likely contributes to Wallerian degeneration. So one of our sources argues that this creates both loss of normal function and toxic gain of function, as the cleavage fragments themselves are sufficient to promote degeneration and may also disrupt CRMP family complexes like the CRMP2, CRMP4. And additionally, after CNS injury, this degeneration-associated pattern appears even proximally. In the optic nerve, CRMP is actually downregulated and tCRMP4 appears, showing failed neuronal degeneration. Overall, the balance between intact CRMP4 and tCRMP4 seems to act like a switch between regrowth and degeneration activity.
Structure elucidation of CMPR4.
The sequence retrieval was from NCBI GenBank, and we had the Homo sapiens CRMP4. For the template selection, we used HHPRED. For the homology modeling, we used Modeler. The AI structure prediction used AlphaFold. For the fold level comparison, we used DALI server. And finally, for visualization, we used PyMol. Before examining the three-dimensional models, the principal functional regions were mapped onto the primary sequence of CRMP4L. The large blue segment represents the conserved core, which is thought to support CRMP family complex formation and includes the region associated with CRMP2 interaction. The red segment represents the possible microtubule-associated region, whereas the green segment may represent the F-actin bundling region. Importantly, the calpain cleavage site is located at threonine 637, potentially at the beginning of the actin associated tail. This positioning suggests that cleavage could directly disrupt normal cytoskeleton regulation while generating the truncated TCRMP4 fragment associated with degeneration.
A homology model of CRMP4 was generated using model with template selected by H. Brad. As expected for template-based approach, the conserved dihydropyridinase-like core is modeled with the highest confidence, especially the beta sheet and the alpha helical region. In contrast, the flexible terminal and low cover region are less reliable. Compared to the other CRMP proteins, the CRMP4 has unique N and C regions. AlphaFold 2 was used to predict the full-length CRMP4 structure without relying on an experimental template. The model shows a compact globular core, which is predictably high confidence, and it is consistent with the conserved CRMP family fold. The PTM score of 0.76 suggests that the overall fold is reasonably reliable at the global level. In contrast, the N and the C terminal regions show low confidence, which is expected for flexible or disordered tails, rather than necessarily indicating model failure. In the structure shown here, this difference is visible directly. The core is compact and predominantly high confidence, whereas the terminal extensions are more extended and low confidence. Using AlphaFold, we can also determine the IPTM, which measures the accuracy of the predicted relative position of the subunits within the complex. But as the protein doesn't have any subunits, this is not necessary, and it was not shown in the AlphaFold.
Awesome. So now we can get into the comparison between the Modeler and AlphaFold platforms. So as you can see on the slide, direct superposition of the Modeler and AlphaFold structures shows strong agreement in the folded core, indicating that both methods recover the same overall CRMP architecture. The structural alignment gave an RMSD of 0.736 angstroms across 485 aligned residues, which supports close similarity in the conserved region. The main differences, though, are outside of the core. So AlphaFold predicts additional beta strand content and an extra loop, whereas the Modeler structure is more conservative, which is expected from a template-based approach. The largest divergence, though, is seen in the terminal and loop regions, where flexibility is higher and structural constraints are less known. But overall, both models support the same conserved CRMP fold, while differing in the terminal regions.
Fold level similarity was assessed using the DALI server with the AlphaFold predicted CRMP4 structure. The strongest hit was 2FTY-A, a member of the CRMP family, with a Z-score of 51.1, an RMSD of 2.2 angstroms, and 470 aligned residuals. These values indicate a very strong structural similarity and confirm that the predicted model adopts the expected CRMP fold. DALI supports the conclusion that CRMP4 retains a conserved ancestral dihydropyrimidinase-like folded core, even though its role has diverged away from the enzymatic catalysis.
The most structurally related protein to CRMP4 is 2GWN. This is a chain of a dihydroprotease found in Porphyromonas gingivalis, a bacteria found in teeth. There's no known function for this protein, but in general, dihydroproteases are in pyrimidine synthesis. It is a zinc-dependent metalloprotease, and it converts carbamoyl aspartate to 4,5-dihydroic acid, and this helps make DNA and RNA. As shown in the picture, CRMP4 is shown in blue, and 2GWN is shown in green. The Z-score is 40.9. This shows extremely high similarity in structure, so they're closely structurally related. The RMSD score is 2.7, and this shows a good structural alignment in superimposed because it's less than 3. It's very interesting to see two proteins that are very, very structurally related. They look very similar, but their functions are completely different.
Here, CRMP2 is shown in light blue and CRMP4 in green. The red and dark blue regions highlight the predicted interface where these two proteins interact, specifically within their core regions. This interaction is important because it likely helps coordinate cytoskeleton remodeling. Since CRMP proteins are known to bind tubulin and actin and influence microtubule assembly, their interaction could directly affect how axons extend and navigate. CRMP4 plays a dual role after nerve injury. In the peripheral nervous system of the injured axons, it supports regrowth, while in the central nervous system, it contributes to degeneration through cleavage mechanisms. So, bringing this back to the interface shown here, the CRMP2-CRMP4 interaction may be part of the mechanism that stabilizes these processes, helps guide cytoskeleton structures during axon extension, while also participating in signaling pathways that regulate degeneration when needed. CRMP2 regulates both major cytoskeleton systems in the growth cone. The microtubule-associated region promotes microtubule assembly, and this activity contributes particularly to philopodia initiation and dynamic rearrangements of the growth cone. The distal C-terminal region between amino acids 470 to 570 mediate F-actin bundling and is especially important for philopodial extension. Loss of CRMP4 leads to smaller growth cones with reduced lamellipodia and abnormal cytoskeletal organization, while overexpression can enhance philopodial growth. Despite this, the structure of the C-terminus tail, which contains the binding site of F-actin, MT site, and T-CRMP4 cleavage site, is unclear. We attempted to model F-actin and MT docking with CollabFold, but the lack of known structure in the binding region made this not possible.
Discussions and conclusions. It is clear that CMP4 retains a conserved dihydropyrimidase-like core due to the AlphaFold HH-pred/modeler overlap and conservation with other CMP4 proteins. But unlike dihydrooroutase, CMP4 does not appear to function like an enzyme, rather a structural molecule. Instead, this conserved core more likely has a scaffolding role, complexing with other CMP4 family members, especially CRMP2. The main limitation of this project is that both models are still only computational, and neither resolves the full biological behavior of CRMP4. The conserved core is supported by both modeling approaches and by Dolly, but the most functionally important region may actually be the C-terminal regulatory tail, which remains much less defined structurally. From this analysis in current literature, it is also unclear how the structure and therefore function of CRMP4 changes with single nucleotide polymorphisms, such as S541Y, or with other post-translational modifications like phosphorylation and sumylation. The most useful next steps would be experimental structural approaches, especially cryo-EM of the CRMP2-4 complex, and solutions-based methods such as NMR or mass spec to characterize the tail. A key feature goal in this area of research would be to compare intact CRMP4 with cleaved TCRMP4 directly, because that could explain how proteolysis changes CRMP4 function and impacts neuronal health. Presented here are the list of sources that we used for this project.